The overall aim of this procedure is to visualize the detailed morphology of Drosophila larval dendritic arborization neurons. This is accomplished by first collecting embryos, aging them, and generating GFP-labeled genetic clones by heat shock-induced flippase expression. The second step is to select larvae with fluorescent neuron clones and take an image of their dendritic arbors. After imaging the larva, they are dissected, fixed, and immunostained so they can be mounted and their dendritic arbors and axonal terminal morphologies can be visualized through confocal immunofluorescence microscopy.
Today I will demonstrate a protocol for morphological analysis of Drosophila larval peripheral sensory neuron dendroids and axons using genetic mosaics. This method can help answer key questions in the neurodevelopmental field, such as what are the genetic programs that control neural identity, dendritic arbor shape, and axonal wiring.
Begin this protocol by collecting embryos on apple juice plates. The heat shock is delivered in a 38 degrees Celsius water bath, and the duration varies according to the embryo genetics and desired clone sizes. For this protocol, generate Markham clones from embryos with a pandendritic arborization neuron driver. Before the heat shock, keep the Markham embryos at 25 degrees Celsius surrounded by moistened tissues for 2 hours. Prepare the collection plates for heat shock by making them submergible using empty plates and parafilm. Then, heat shock the Markham embryos for an hour to generate small clones or single neuron labeling. To increase the number of clones, use an extended heat shock by incubating for 45 minutes in the bath, followed by 30 minutes at room temperature, and then another 30 minutes in the bath. To generate flip-out clones, collect embryos for 24 continuous hours. This protocol utilizes a Class 4 specific driver. Heat shock the flip-out embryos for an hour. After the heat shock, place the collection plates in a 10 cm petri dish surrounded by moistened tissues and culture the embryos at 25 degrees Celsius. During embryo culturing, monitor and replenish the yeast paste as needed because nutrition critically affects neuronal development. Maintain the culture until wandering third instar larvae can be collected. Briefly and gently, rinse the third instar larvae in tap water and transfer them to an agar plate. Now, examine the larvae using a powerful fluorescence dissecting microscope and insect forceps. Identify larvae with GFP-positive neurons in the body wall and transfer them to a depression slide with a drop of 80% glycerol. Place a cover slip on the slide to immobilize the larva. Ensure that no air remains between the larva and the cover slip. Use standard confocal microscopy to visualize the neurons of interest through the larval cuticle. To change the larva's position for an improved viewing angle, gently push on the cover slip to roll the larva around. Begin this protocol by transferring a larva to the sill guard plate using insect forceps. Then, pin it down at the anterior and posterior ends. Then add calcium-free HL3.1 buffer. Now, cut halfway through both ends of the larva, perpendicular to the anterior-posterior axis. Then, cut along the midline between the two trachea. Carefully cut each individual tracheal connection to the body wall before gently removing the gut. This keeps the segmental nerves running between the PNS and the CNS intact. With the larva still pinned to the sill guard plate, fix it in 4% paraformaldehyde in PBS for 20 minutes at room temperature on a gently rotating shaker. Perform each dissection in less than 5 minutes, otherwise the dendrites of the dendritic arborization neurons will begin to degrade. After the fixation, remove excess larval tissues. Then, wash the fillets 3 times with PBST for 10 minutes per wash. After the washes, incubate the larvae for 20 minutes at room temperature in blocking solution with gentle shaking. After the block is complete, replace the blocking solution with the primary antibody solution. Place the sample in a small plastic container surrounded by moistened tissues and incubate it overnight at 4 degrees Celsius. The next day, continue the incubation for an additional hour at room temperature. Then, wash the larva fillets 6 times in PBST for 10 minutes per wash. Now, add the secondary antibody and put the fillets in a dark container to prevent photo-bleaching of the fluorophore. Incubate the fillets for either 2 hours at room temperature or overnight at 4 degrees Celsius, followed by 1 hour at room temperature. Finally, wash the fillets 6 times in PBST for 10 minutes per wash. The tissues are then ready to be mounted. To visualize axon termini, simply mount the larval fillet cuticle side down in 80% glycerol in a depression slide. To make clearer images of dendrites, make a permanent mount with PLL-coated cover slips. To coat the cover slips, thaw an aliquot of PLL, bring it up to 10 ml of double distilled water and add 20 microliters of Kodak PhotoFlow. Submerge the cover slips for 30 minutes in the PLL solution and air dry them upon removal. Then, briefly rinse them with water and air dry them again. Repeat this process twice, beginning with the PLL bath. The treated cover slips will last approximately 1 month. Now, mount a dissected larval fillet muscle side down into a drop of PBS on a PLL cover slip. The fillet will adhere quickly to the cover slip. Using a piece of tissue paper, remove as much liquid as possible. However, do not allow it to completely dry out. Next, dehydrate larval fillets in a series of 10-minute ethanol baths, beginning with 35% ethanol, followed by 50%, then 70%, then 95%, then 100%, and finally a second 100% ethanol bath. Follow the ethanol baths with 2 or 3 10-minute baths in xylene. Then, put a drop of DPX on a clean slide and carefully set the cover slip into the DPX, fillet side down. Keep the slide in the dark and wait 1 day for the DPX to set before imaging the tissue. Using in vivo confocal microscopy, it is possible to capture images like this, the entire arbor of a class 4 dendritic arborization neuron. This is a close-up of a dendritic arbor from an IHC labeled class 3 neuron that has been correctly fixed to preserve morphology. The inset shows the degradation that can occur after an unsuccessful dissection and fixation. This overlay shows a single class 4 axon terminus in the CNS. The axon is labelled with anti-GFP in green, and all class 4 termini are co-stained with anti-CD2 in magenta.
While attempting this procedure, it is important to remember to dissect and fix samples quickly. When tracing axons from the periphery to the CNS, it is useful to keep in mind that PNS neurons in each body wall segment project to the cognate segment of the ventral nerve cord. Also, if you only want to visualize dendrites, remove the CNS during dissection and staining can be carried out in Eppendorf tubes instead of on plates. When mounting these fillets for dendrite imaging, first cut off the head with mouth hooks and posterior with sparkles for a flutter preparation.